Here is a diagram of the structure of the cell membrane. We have the protein channel and protein pump, both of which are integral proteins. Integral proteins are a type of membrane protein that is permanently attached to the biological membrane. However, peripheral proteins are membrane proteins that adhere only temporarily to the biological membrane with which they are associated, such as immobilized enzymes.
In between the proteins, there are the phospholipid heads and tails. A phospholipid is a lipid containing a phosphate group in its molecule. The heads contain a negatively charged phosphate group and a glycerol. Also, they are hydrophilic, meaning that they have a tendency to mix with, dissolve in, or be wetted by water.
Between the heads are the phospholipid tails, which usually consist of two long fatty acid chains. They are hydrophobic and tend to repel or fail to mix with water. The heads and tails make up the phospholipid bilayer, a thin polar membrane made of two layers of lipid molecules, the heads and the tails. The bilayer is considered amphipathic because it contains both hydrophilic and hydrophobic parts.
Finally, in between the phospholipid bilayer is the cholesterol. Cholesterol is a compound of the sterol type found in most body tissues, including the blood and the nerves. Cholesterol and its derivatives are important constituents of cell membranes and precursors of other steroid compounds, but high in concentrations in the blood, mainly derived from animal fats in the diet, are thought to promote atherosclerosis. The chain coming off of the protein on the end is the carbohydrate chain.
Now we are going to talk about the functions of membrane proteins. First are hormone binding sites, or hormone receptors. An example of this is an insulin receptor, and the function is performed by integral proteins, and they interact with the receptor to modulate activity.
Next, immobilized enzymes with the active site on the outside, for example in the small intestine, are performed by peripheral proteins. An example is a natural polymer derived from the cell wall of some algae. Calcium or magnesium alginate is a most commonly used matrix. They are inert and have good water-holding capacity.
Next are cell adhesions, which form tight junctions between groups of cells and tissues and organs. This is integral. An example is cadherins, a family of Ca2-dependent CAMs, are the major molecules of cell-to-cell adhesion and play a critical role during tissue differentiation. The most widely expressed, particularly during early differentiation, are the E-negative, P-negative, and N-negative cadherins. The brain expresses the largest number of different cadherins, presumably due to the necessity of forming very specific cell-to-cell contacts.
Another function is cell-to-cell communication. For example, receptors for neurotransmitters at synapses, where neurons continuously gather information about the internal state of the organism and its external environment. They also evaluate this information and coordinate activities appropriate to the situation and the needs of the person. A real-life example is a nerve impulse, which is the transmission of a coded signal from a given stimulus along the membrane of a neuron. There are electrical and chemical impulses, and they are both integral and peripheral.
We mentioned protein channels earlier, and they are channels for passive transport to allow hydrophilic particles across by facilitated diffusion, which is integral. The channel is a protein that allows the transport of specific substances across a cell membrane. The process by which molecules pass through a membrane via a channel protein is called carrier-mediated transport.
Another part we mentioned earlier were the protein pumps. They use the energy of ATP hydrolysis to move ions or small molecules across a membrane against a chemical concentration gradient or electrical potential. This process, referred to as active transport, is an example of a coupled chemical reaction.